Chapter 14, Section 12, Sulfate and Sulfur Reduction. Okay, we're going to be looking at here more inorganic metabolic methods for production of ATP. Here we're going to be looking at how some prokaryotic cells can utilize sulfur. Now, inorganic sulfur, so this isn't a thiol functional group attached to a carbon skeleton. This is a compound where sulfur is the central atom, that everything else, the hydrogens, the hydroxyls, whatever, the oxygens, are attached to. What we're looking at here is the reduction of, sulfate to hydrogen sulfide. So these are going to be bacteria that are commonly referred to as sulfate-reducing bacteria. Some of these can be obligate anaerobes. Others could be assimilative. They can take in the sulfate and use it for, biosynthetic purposes. Remember, amino acids have an amino functional group. And some of them, so there's, the previous couple of sections where some of that nitrogen can come into play. Others are going to have an R group that has a sulfur. Cysteine, methionine are two amino acids that have sulfur as part of the R group coming off of the alpha carbon. You can also use sulfate as an electron acceptor. And we're going to be looking at pathways utilize this. Now, when you go and you look at, again, organic sulfur here, this thiol, the sulfate group, the sulfur group is a functional group. Sulfide, thiosulfate, sulfate, sulfite, sulfur dioxide. As we go through, we see that the oxidation state. What energy state is this sulfur atom at? Changes depending upon. So when you're going from one to another, how far apart are they? It also tells us how much energy could potentially be there. The sulfate is energetically less favorable than O2 or NO3. So our energy yield here has the potential to be lower than what we would see with normal aerobic metabolism and less than what we've seen previous section with nitrogen metabolism. Hydrogen is going to be an electron donor here, okay, because remember hydrogen is nothing more than an electron with proton baggage. Some species can use things from their environment, lactate, pyruvate, from other organisms, acetate, long-chain fatty acids, whatnot. Because think about it, long-chain fatty acids are long chains of carbon with hydrogen there, so they're going to strip it off. And somewhere in the process, sulfur is going to be either the final electron acceptor or something like that. The reduction of the sulfate to hydrogen sulfide is going to require 8 electrons, and it's a multi-step process. So instead of giving up electrons that can be utilized for an electron transport chain, here we're going to be using some of them. We're also going to have to bring in some ATP to form an intermediate known as ATP sulfurylase, adenosine phosphosulfate. ATP sulfurylase is the enzyme. Enzinase, the enzyme. ATP sulfur, adenosine phosphosulfate. We also see that this sulfur metabolism, depending upon the microbe, can be a dissimilative or assimilative. Are you breaking it down and things like that completely, or are you going to break it down to a certain point and then start utilizing it in your own organic compounds? Here's that adenosine 5-phosphosulfate. adenosine. The adenine nitrogenous bases are up here. They're not shown. They're just written here. Here's our 5' carbon, our phosphate. So it's a nucleotide, and then a sulfur-containing compound. You could also end up with, so that's APS, and then you can also have PAPS. PAPS comes into play, phosphoadenosine 5-phosphate. So 1, 2, 3, 4, 5 carbon. So it's 1, 2, 3. 3 carbon has a phosphate, phosphoadenosine, and then the 5' carbon phosphate sulfur that we see here with APS. Why is this important? Well, SO4, S1, 2, 3. The fourth one was there already with inorganic phosphate, or it came in with the sulfur and then the inorganic phosphate one gave up. Chances are, since you're starting with ATP, and here you see the beta and the gamma phosphate are being released, that these are going to be complete. So this is the oxygen coming in with the sulfur. Makes sense biochemically. Sulfate to APS, APS to PAPS. Cool. So APS and PAPS are intermediates that are going to be utilized in other places. APS can then be, APS reductase. You cut off, release the AMP, and you now have sulfite. Sulfite becomes hydrogen sulfide, which can then be excreted. You probably saw this in the lab when you last lab when you went and you looked at your Sims agar, and you noticed that E. coli had turned it black. It went through, did the sulfate that's in the agar, turned it into APS to sulfite to hydrogen sulfide. The bacteria, they're going to make the sulfate to APS and then to PAPS. Again, sulfite, hydrogen sulfide. And this is going to be then assimilated into, say, like amino acids or anything that may need a sulfur atom in it. Dissimilative, assimilative. Biochemically, how's this working? Well, cytochrome Cs are going to be important. C3 is our cytochrome C of choice here. Takes the electrons from several different dehydrogenases, and you're going to get hydrogen. It's either going to use it or produce it depending upon which way things are flowing. Sulfate reducers that you are using organic substrates. So basically it's some form of an organic molecule that has a sulfur on it. So it's maybe one of those amino acids. Well, it's going to oxidize them and then produce hydrogen gas that will then feed into the electron transport. Dissimilative that are breaking down are going to use a flavin-based electron bifurcation. We talked about that in a previous section, where things are going to be broken down, one way, and then splits can go, some of it goes this, some of it goes there. Exactly which ones here we'll talk about on the next slide. But basically what we're looking at is a balance of flow. Balanced flow, they call energy converting hydrogenases. High levels of this means it's above a threshold, things go one way. When that drops, then it goes a different way. So this way we keep things in check. So when you go and you look at outside the cell, inside the cell, things are color-coded. Common, hydrogen only, growth on lactate only. No, I'm not going to go through all of this because this is a messy, messy thing. What I want you to focus on is hydrogen gas, H2, 3 free hydrogens outside. H2A's, passes on electrons to C3 here, which is going to go to an abridged cytochrome complex. Boom, boom, electrons pass through here, all these proteins, pumps hydrogens. The electrons that are taken from a ferredoxin, reduced, oxidized, fed here so that sulfate turned to APS, sulfite, boom. This here can be, added to and turned into hydrogen sulfide, which will be excreted. Boom, again, reduce, 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 pass on. So this mess right here is all about taking electrons from hydrogen gas and then passing them through, allowing the pumping of hydrogens, and then those not quite as excited state electrons are going to be used in the sulfate to APS, sulfite to hydrogen sulfide. Ferredoxin, ferredoxin, ferredoxin. Lactate to pyruvate, the pyruvate to acetyl-CoA to acetate. In the process, you get, substrate level phosphorylation. Do I expect you to memorize this? Holy shit, no. This is a lot. This may actually be too much for one figure, but it is trying to show you all the probable ways and configurations you could see where hydrogen is serving as the primary electron donor. In the process, that electron pumping hydrogens and then is going to finally be used to reduce. So we can go from sulfate to hydrogen sulfide. Keeping in mind that this hydrogen, this sulfur metabolism, maybe the electron donor isn't hydrogen. Maybe it's something else in the environment. Hence that lactate and the pyruvate that were shown there. The pyruvate could be from the cell's own glycolytic pathways, or it could be pyruvate that was excreted because it was overproduced by some other cell. Who knows? Lactate, the sugar lactate, depends on which bacteria this is, what environment it's in. It could be an environment with lots of lactate. It could be a bacteria that's in an udder of a cow. I don't know. But its environment has, intermittent lactate, availability. Okay. As we saw in the last, that means that they're going to be the ones that are giving up the electrons that are then going to be utilized for the reduction of sulfur. You see that marine sulfate reducers are going to also, utilize some acetate oxidation. Remember, acetate is a 2-carbon thing. So there's no way they're going to be the presence of lactate or pyruvate in a marine environment, but depending upon what type of sulfur vent or whatever they're around, maybe there's other things present. If they happen to be an autotrophic microbe, okay, they can use this sulfate reduction as long as the hydrogen acting as an electron donor and an acetyl-CoA pathway that we've talked about in previous sections. So there's their mechanism for carbon fixation. Other potential means of utilizing sulfur reduction, when they go through and they do this, there's alternate mechanisms because they need to conserve. This sulfur metabolism is a low energy yield, so if there's other things that can be used, they will use, and then this is there when they can use it. What we see is just how, as with cellular metabolism, you always seem to have this balance of exergonic, endergonic. Well, turns out sulfur reducers always seem to couple with, coexist with sulfate reducing. Sulfate reducers overproduce sulfur-containing compounds, which they excrete out, which these guys then take in and further reduce, ending with hydrogen sulfide. There are some that do a disproportionation. Okay, they overproduce one over the other, so the oxidation reduction, to form two different products, which can push and pull on the metabolism one way or another. Desulfivibrio sulfodismutase, thiosulfate, hydrogen sulfur, hydrogen sulfide, and sulfate. Okay, that's a bit weird. But it takes the thiosulfate, turns it into hydrogen sulfide and sulfate disproportionately, which means that its metabolism is a bit weird. I'm not going to go into it too much. We're just going to mention it here and move on. And then there's phosphite oxidation. So phosphite, which is, anaxonic degradation of phosphorus-containing compounds. They're going to utilize this phosphite in a sulfur reduction. Example of this is disulfotignum phosphatoxidants. Wow, those are some words.